Hello everyone. This is Dr. Atul Pradhan, Assistant Professor, School of Agriculture, Sage University, Bhopal. So, in continuation with the previous two lectures on plant genetic resources, today we will discuss about the third part of plant genetic resources, that is depend on the gene pool concept as well as seed collection. So, in these two topics, we will go one by one that what is gene pool system, what is various types, and the second part that is about the various types of seed collection. So, before going to this types of gene pool, we have to know that what is gene pool or what does it mean. So, it is the set of all the genes or the genetic information in any population of a particular species. Means gene pool, it is having the total diversity or it is having the total genetic variation of a crop which include all the cultivars, which may be absolute cultivars, which may be wild relatives, which may be wild species. So it is having the sum of all the cultivars which are available for the breeding use. So that is the gene pool. So this gene pool concept initially was given by Harlan and DeWitt in 1971 for the effective and judicious use of germplasm. So as in the definition, this gene pool indicates the total diversity. So what is the reason behind this gene pool? So, in the past, huge pools of various wild and indigenous breeds have collapsed, causing widespread of genetic erosion as well as the widespread of genetic pollution. So that's why conservation is important. So in the view of gene pool, from the gene pool we can easily conserve all this genetic material in a particular type of conservation. So this is about the total gene pool. So based on the concept of hybridization as well as compatibility study, this gene pool can be classified into four types, which is primary gene pool, second one that is the secondary gene pool, third one tertiary gene pool, as well as the last one that is the quaternary gene pool. So we will discuss one by one that what does it mean. So first of all, we will go with the primary gene pool. So in case of genetic material, when intermating is easy and leads to the production of fertile hybrids, it is known as the primary gene pool, which is also designated as GP1. So in this primary gene pool, the plants of same species or the closely related species which can produce completely fertile offspring on intermating with each other. So that's why this primary gene pool or the GP1 is of the prime breeding importance. So if we consider about the example, so the cross between the two sunflower varieties of the same species, that is about the GP1. So in case of GP1, it includes two subspecies. The first subspecies, that is subspecies A, which is having all the cultivated races, and in case of subspecies B, which are having the spontaneous races, which may be wild or breed species, with which the cross can be compatible and can produce the fertile offsprings on intermating. So this is total about the primary gene pool. In case of secondary gene pool, in case of genetic material which leads to the partial fertility on crossing with the GP1, that is the primary gene pool, on crossing with the primary gene pool, it is known as the secondary gene pool. So in this case, the plants that belong to related species but which have some of the barriers on crossability with the crops, right? So this in case of genetic secondary gene pool, this transfer of gene from such material to the primary gene pool or GP1 is possible, but difficult, and it leads to some of the points which maybe leads to the partial sterility or weak hybrid, which may leads to the chromosome pairing poorly or not at all, which may leads to the recovery of desired phenotypes. The phenotypes can be developed, but their recovery may be difficult in the subsequent generations, and the gene pool is available to utilize by the plant breeder or genetics with due effort required. So these are some of the points. That's why this GP1, it is producing the partial fertile or partial hybrids, and it is causing some of the difficulties. So these are the various difficulties which causes in GP2. So, one of the example in GP2 that if we are crossing with Agilops tauschik with Agilops peltoides in case of wheat, so we can get the hybrids, but they are the partial fertile. So this is about the GP2. The third one, that is the tertiary gene pool, are the GP3. So in this, if genetic material which leads to the production of sterile hybrids on crossing with the GP1. If we cross this GP3 genetic material with the GP1 primary gene pool, which leads to the sterile hybrids, that is called as the tertiary gene pool. So this tertiary gene pool results in lethal or sterile hybrids due to abnormality in development of embryo. For example, if we cross with that triticum turgidum with Agilops peltoides, so definitely we will get the sterile hybrids. So this is about one of the example. Another example, that is about the triticale. So in triticale, if we cross the wheat with the rye, we will get the triticale, that is the man-made cereal. But this cereal developed by considering some of the special techniques, which may be embryo culture, tissue culture, chromosome doubling, or using the bridge species. So only with the special techniques we can get the fertile hybrids. So in with the help of special techniques, transfer is possible. Uh, so this is about the tertiary gene pool. The next gene pool, that is one of the new type, that is called as the quaternary gene pool or designated as GP4. So this new type of gene pool GP4, it is also referred to as the gene ocean, has thus been made available by the modern science. So it is beyond the limits of the gene pool, beyond the limits of the human imagination. So in this quaternary gene pool, some of the special techniques we are using of recombinant DNA technology. So one of the best example, that is the transfer of Bt gene, that is the transgene from the bacteria Bacillus thuringiensis in cotton or in maize or in tomato and other plant species. So this is about the quaternary gene pool, and it is required some of the special techniques in case of recombinant DNA technology. So this is one of the picturization of the modified gene pool concept in plants based on the hybridization study, which is having GP1, GP2, GP3 and GP4. So some of the key points we have to remember, that the most commonly used gene pool in breeding program, that is the GP1. In case of GP1, we can get the completely fertile hybrid seeds. In case of GP2, we are getting the partially fertile seeds produced from crossing with GP1. In case of GP3, great difficulty we are having in case of hybridization and produce sterile hybrids when crossed with the GP1. This GP4 is also called as the gene ocean, and transgene, the term is related to only the GP4. So these are some of the key points we have to remember in case of the gene pool. So this is about the gene pool concept. The second concept, that is about the various types of seed collection. So on the basis of use and duration of conservation, this seed collection can be classified into three types, major three types, that is the base collection, active collection and working collection. So in case of base collection, in the name itself, it is also known as one of the principal collection or the basic collection, which refers to the whole collection. And this includes a maximum or more number of accessions present in all the germplasm or plant genetic resource of a crop. This base collection can be stored or conserved up to 50 years or more than that, having the temperature of minus 18 degrees Celsius or to minus 20 degrees Celsius with 5% moisture content. And this base collection only disturbed when they required for regeneration. And these are used only when germplasm from other sources is not available for use in breeding. So in uh, mm, less cases, this base collection is required for the breeding purpose. The second, that is the active collection. So this category of germplasm is actively utilized in plant breeding program and are conserved for medium term, that is for 8 to 10 years or up to maximum 15 years. This active collection games, we can store them. After that, this collection are stored at 0 degrees Celsius with moisture content of 5 to 8%. And definitely in the name itself, these collections are actively utilized in the breeding program, and their germination test is carried out after every 5 to 10 years to assess the reduction in the seed viability. So this is about the active collection of various plant genetic resources. And the third one, that is the working collection. So this working collection is frequently used by the breeders in the crop improvement program. And in case of storage, they can store it for short term of 3 to 5 years with having 5 to 10 degrees Celsius with moisture content of 8 to 10%. So these are the various types of seed collection, that may be base collection, active collection, and working collection. So there is another category of seed collection that is called as the core collection. The core collection means in case of best collection, if it is not possible that we can uh, represent the best collection at a one time. So instead of this, we are using one of the subset of the best collection. So that subset is called as the core collection. So it refers to the subset of a best collection which represent the large collection or base collection. So this is about the core collection, and this concept of core collection was proposed by Frankel in 1984. So this is the brief information about the gene pool concept, various types of gene pool, and the various types of seed collection which are working in this plant genetic resources. So this is it. So thank you and stay tuned and subscribe to Sage University book. Thank you again.